Hello, everybody. Can somebody online give me a sign that you can hear us? That's very good. Thank you, Johanna. Okay, so this is the last session, and unless I don't know, I haven't seen what's his name, Eligio here. So he's not online. Did he present over there?
Yes,
I will
here.
replace. Very good. Okay, so then we have 4 talks. And I think the first one is Agrobiodiv. And let me start the presentation. Good. So, Maria.
Okay, thank you. So going back to this apple biome problem, the project, the title is Microbiome and Genomic Analysis in Apple Germplasm towards broadening genetic resources to breed for resilience varieties. So with this title, I think it's very comprehensive. But let me explain you more about the story, why we applied for this project. Is because the apple production is facing interesting, important challenges due to the varying environmental conditions, diseases, and management practices. So the climate change and modern agricultural pressures demand more resilient crops. So we think that a deeper understanding of the interaction between the genome, the environment, and the management could help to ensure sustainable apple production. So why we decided to focus in the microbiome concept? Well, we know that the environment determines the microbial composition, the community composition in the orchard, in the plants. We also know that there is the host genetics may help to the recruitment of beneficial microbes, and also that these microbes may determine how the plant responds to some challenges.
Yes, we can. Do you hear me? We can hear you, but I don't know how about the others.
Hello?
But I hear the background way louder than the actual presentation.
No? Okay. So considering all this, we believe that the microbiome in the plant deserves special attention. So the question is, what if we could predict resilience? What if we could predict which apples genotypes will perform better under specific environmental and management conditions? And what if we could manipulate the plant microbiome to boost resistance to some diseases? So you can imagine the impact. These insights could help apple breeding and agricultural management to ensure food security and reducing chemical inputs. So the hypothesis in this project, well, we have several hypotheses, but I think the most important and understanding how plant genotype controls the microbiome composition may help to understand the disease resistance and stress tolerances, and also if we can use this knowledge, this microbiome, we could develop solutions, sustainable solutions to improve apple crop performance. So with this idea, the objectives, we don't have many results now, so I want to focus this talk in the questions that we had when we started this project, because I think it's what's important at this step. But the questions, what some of them were. How do different environmental conditions affect the plant traits, the crop traits, the growth, the phenology, the productivity in apple? What are the genetic determinants that influence some of these traits across environments and with different managements? And can genomic prediction methods be developed to forecast apple performance and disease resistance? So to answer these questions, we rely on a very, very nice and unique tool that is the Apple Red. This collection was created as an output of a European project and has kind of 500 genotypes that are planted, that grow in 6 European locations since 2016-17. And since then, they have been phenotyped for multiple traits. So this collection of 500 genotypes, you can see here, they are planted in these European locations in 2 management conditions. The whole collection is management 1, it's in commercial standard management, and the second more reduced sample of the genotypes, 200, are in low input management. So see, in the apple biome, we are analyzing these materials all the way evaluating for disease resistance or tolerance or disease incidence. Okay? So far, our colleagues from Angers have been working on putting together and developing a method that's called WISER. That's a novel approach for more accurate phenotypic estimation. So because we are having data from different places of the same materials, and so a way to automatically treat all this data, removing outliers and calculating and putting all the data together, it's very important. And so in this WISER also helps or can be used to visualize genetic and phenotypic data structure and also to evaluate the genomic prediction accuracy. So I'm going to show you just one of the examples of the results. And it's how, if we use this WISER treatment of the data, what is the, for example, the genomic prediction, how it's improved the genomic prediction of flowering time. So you can see here, like this, you have, when in this here is the genomic prediction ability when we use this WISER methodology in the models, in the other models, right? So comparing, this is in flowering time, but there are examples in many other traits. So this will be, it's already in a GitHub repository, the scripts that will be made available after publication, hopefully soon. We have more questions. So, for example, how the plant genome influencing in the composition, in the recruitment of some microbial taxa. We are talking about the phylosphere. We are not working with the soil. We are working in the fungal, not in the bacteria, because some reasons that they can explain you later if you're interested. What is the relationship between genotype, environment, management practices, and the microbial diversity in the phylosphere? Also, which microbial taxa are associated with plant resilience, if any, and which of these key microbial hubs correlate with phenotypic traits? So, well, these are some of the questions that we have, and for that we designed the big experiment that consisted of taking leaves from the materials of the apple red pod. So management 1 and management 2, we are talking about more than 4,000 leaf samples. We didn't have enough with that, so we said, okay, let's go to monovarietal orchards, and we increased the sample size to almost 5,000, 4,800 samples. But I tell you that it's a lot, and it was really a challenge. Why it was a challenge? Because, well, it's, the lettering is a bit out of size here, but it was a challenge because some of the partners didn't have enough structure to lyophilize, to freeze dry all the samples together. This is a very big one that we had here that it was for animal farming, so we could put here our samples, but in other cases it took longer to dry the samples. but we did it. So we decided the protocol in which is the moment of the decision to collect it after some days of training and some days of the treatment. So we coordinated everything and it worked very well. So we can congratulate each other for that. And then the second challenge was that, okay, this is a lot of material. It's going to be very expensive. So we cannot just try and see. So we needed to find a cheap, fast, and reliable method for DNA extraction. And we needed to find a good method for sequencing. So for the DNA extraction, we decided to test kits. The kits were the fast, but not the cheap, and the reliable, but not the cheap. And a method that we use in the lab for Apple that it works very well, and it's also described a word for fungal or microbiome composition. So it's an English method. I don't have here the reference. I can give it to you. So when we did the IPS2 amplification, and what we found is that the no kit was okay, but it was not very reliable between different sample sets. And one of the kits, the NC-Tech, was the one performing better, giving more number of OTUs, while the Quiagene that is regularly used in these protocols was okay, but it was not the best. And actually, we got a very, very good quotation for NC-Tech. So everything fits. And you can see here that also even having the larger number of OTUs, then we had even more depth with this method. So it was perfect. It was a bit more expensive, completely more expensive than the English, but working much better. What we found as we were amplifying the Apple DNA in detriment of the microbial DNA. So we already added a blocker of the DNA as in a protocol that we found and a collaborator also used it. But we decided to start a second pilot analysis. And in that case, we used the blocker in different concentrations. And actually, we wanted to know if the blocker was working. So we did one without blocker, one with the concentration that was in the bibliography, and one increase in this concentration. And actually, it worked. It worked very well. And we, at the end, could have much abundance of the taxon. So it took us sometimes some delay on the previous, on the schedule of the project, but we wanted to have this data very clear. So now what we have to do is the sequence the whole sample set with this methodology. And for us, a challenge has been to analyze all the computational data, all these large amounts of data, because even if it's the IPS, we are sequencing it with Illumina, so we have a lot of data, and we have some problems finding a profile for that. So we have started a collaboration with Sequentia Biotech. They are going to do as collaborators some of the analysis and part, or at least at lower cost. So at the end, we want to identify the genotype, environment, and management of specific microbial community. Also, the interaction with the genotype, and also find which part of the genome participate in the recruitment of the beneficial microbes. So with that, we can use these materials, these alleles, for breeding. So next steps, analyze, dissemination, of course, and meetings with the stakeholders. Now I think it's the time to explain them what we want and where we are, and to make them participate more in the project. Although in this project we are researchers and also companies. So there are producers and breeders in the project. And of course, identify genotypes for breeding. It's Apple. We are not going to use this material for at least to see results in the project, but we can make plans for processing in each project. So the question, I think the big question is, what if we could manipulate the plant microbiome to boost resistance by breeding or by adding some products? It's something that long-term objective, but it's a question that, or one of the objectives here. So, okay, I want to thank the collaborators. As I said, we are industry partners that are companies, breeders and producers, and the academic partners. the posters of the Apple Red Pop. So it's a very nice tool that it helps us to build consortiums for different projects. And also we want to thank Sequentia because as a new collaborator is helping us to do the analysis. We have a web, we have also a Twitter account that I didn't check here, but you are welcome to look for us in there. So if you have questions.
Yes, if you have questions.
Yes, can you comment a bit more on how this wiser process works?
I cannot comment much more. Here I see Elaine, that is the leader of this group that was here. You can ask her if you want, I can give you the contact. It's curating the data, removing outliers, doing some correlation. But also, if you see the link, I'm not sure if it's here. Maybe you can find something here. I have to. You can check. But otherwise, I will give you the contact of Helen because she can explain you much more of this methodology.
Maybe I don't know if I got it. How are you going to link the GWAS analysis with the microbiome analysis?
Yes, we are going to use different approaches. But one is to put select the abundance of the taxa within as phenotypic values, phenotypic data. So just use these values, the abundance of some taxons with the GWAS analysis, or also some correlation. Yeah,
And one idea would be that particular phenotype that you observe within the population of trees of apple
Sorry.
One idea would be that one of the phenotypes that you study within the population of apple trees will be associated with a particular microbiome composition.
Yeah, we are also going to study these correlations. If we find a correlation between some of the taxons with the traits. I think it's more than with the traits, with the phenotypes. I think would be more correlation with the disease resistance and abundance. That's something that we need to check. Also, there are more things in this project. Then we will validate the results with another sample set in a different year and see probably more along the season. But the big part of the project is that. And the point here is we have different contrasts, right? It's the genotype, the environment, the management, and all together and different combinations. So that's the complexity of the analysis, and that's why we wanted to have someone able to really analyze the data together. That would be like you do RNA-seq analysis, that you have different contrasts and everything. You can imagine that when we are going to check environment, for each genotype, we have it in 6 environments, but in each place we have replicates because there are 500 genotypes of replicates and management. So at the end, we are going to work with a large data. Yeah.
Kim, I don't want to be impolite, but I think we need to get going.
Okay.
Otherwise,
Okay, that's a very simple question.
Okay.
So
I need a quick question, a quick
we know that endophytic microorganisms such as bacteria and fungi colonize the plants when they first begin to grow. Do you think to study the endophytic fungi, you work only on phyllosphere?
Sorry?
You didn't work on endophytic bacteria or fungi. Have you been study on it?
We will study only the phyllosphere, that's all, the surface also and the mesophag.
No, I'm talking about the endophytic fungi and bacteria.
We are going to see what we have there.
That's very important to
and we will check which are the endophytic and which are more. Sure, we are going to find some that are diseases.
Okay.
So we will check what we have. We don't know yet. But
Good.
sorry for.
No, that's fine. So we continue with the project in.
Well, hello everyone. Like I said before, my name is Cassandra. I work for the Swedish University of Agricultural Sciences in Uppsala, and I'll give this presentation about improving agrobiodiversity for resilient pest control services across landscapes. And my colleague Eva will tell you something about the social aspect of this project. I hope to keep your attention a little bit longer. There are cool detailed pictures. All right, so just to give you a little bit of background, as I explained in my pitch, there are these various aspects that affect the diversity of predator communities that provide pest control to our crops, such as the diversity on the farm level and the management by the farmer, the surrounding landscape, and also the climate. And even though these various aspects have been studied quite a lot in the past, what they do to predator communities, a little bit less attention has been put on the resilience of the function provided by these predator communities. So this is the gap that our project aims to fill. It's still very much ongoing. All right. So one way in which we try to, define resilience is the ability of a system, so in this case, the pest control system, to recover or maintain its function after a disturbance. And this disturbance can be human-induced, for example, by plowing a field, or a little bit more natural, for example, a fluctuation in weather. And two ways in which biodiversity or the diversity of species contributes to resilience is through functional redundancy. So here we have an example from work by my colleague Benjamin Veit. We have different predator species on the x-axis and their climate niche on the y-axis, and functional redundancy is basically when multiple species can contribute to the same function under the same conditions. So if we take the mean temperature of 24, there are, for example, several species that contribute to function at this time. And another way in which resilience can be enhanced is by the diversity of response to a disturbance by the different species. So even, for example, if the temperature were to rise, there might be other species that are active at a higher temperature and still maintains this function over time. So to investigate this further, we introduced the IMPRESS project here. It's a project across 5 countries where Lithuania is especially contributing the social science aspect, and the ecology aspect is happening in Sweden, Germany, Austria, and Italy, representing 4 different climatic conditions. And in each country, we replicated the study in various landscapes, from complex to simple, where we assume diversity is high in complex landscapes and low in simple landscapes. And we compare paired fields in each landscape, one which is plowed before sowing, one where conservation tillage is used, where we assume that the effect on biodiversity is lower. And the work we have done so far in the various work package is, first of all, to assess the diet niches of these different predators. So we collect live predators from the field, we make them throw up, and we extract the DNA from their puke to assess which animals these predators are actually eating, which ones are responsible for the pest control in our fields. And if the poor beetles survive this ordeal, we can release them back into the environment, so we don't have to kill them for this. Secondly, we also looked at the community composition after a certain disturbance. So we did measurements before and after disturbance, in this case, the harvest in all 4 countries. And we tried to see how do the communities recover after certain disturbances apply, and how does the landscape and the management of these fields affect this. Additionally, we also want to do more research on the climate niches of predators and predation, and we believe this to be a very important aspect of the project, because with increasing and changing climate, this might be something that affects resilience quite a lot. So in order to do that, we do very short-term tracking of only 2 hours at a time, and measure temperature and humidity during these time windows to see under which conditions are the predators actually active and predating on various prey. So on the left, right for you, hand picture, we see the larvae of a lady beetle eating an aphid that is glued on a card So we can basically measure exactly under which conditions does the predation of pests actually happen in field conditions. And additionally to this, we also have mesocosm experiments done especially by our Italian partners, where we basically want to validate the findings that we have in the field and expose various species to various climatic conditions. And especially we want to try to disentangle the effects of temperature and humidity, which are very closely correlated out in field conditions. And this here is a video, but I don't know if it's working. No. Oh well. You just have to imagine that in this video, you see the beetle consuming a prey. Go for it. All right. So Eva will tell us something about the social science aspect of our study.
If you allow me, I stay in my place. So we have like a little social project next to this big, interesting ecological project. We want to look what we can do to encourage the farmers to use more sustainable practices to improve this resilience for pest control. For the moment, we are finalizing where we look which actors work in this field, let's say, and we base on this Abbott and Snittle idea of governance triangle. We also do this regulatory documents and analysis and some interviews with experts from these different corners of governance to assess what is the current regulatory framework and practices. And the last step will be to clarify these supporting factors and barriers. So we kind of expect to draw concern profiles of the respondents and to see when farmers tend to be more aware and tend to practice more sustainable and natural pest control, if you can say it. And in the next slide, some very little, some initial results. What we really see that at the scientific level, there are views that natural control refers to the control of insects provided by nature, by natural processes, so without human involvement, and biological pest control refers to cases where people purposely manipulate populations of natural enemies, let's say. But in the documents, we don't have any definitions, neither of natural pest control or biological pest control, but certain legal acts mention both in the context of integrated pest management. So what also we see that member states kind of possess legal tools to implement natural pest control under the directive of sustainable pesticide use. Within this integrated pest management, however, we see that it's very limited and no progress was made. Countries are obliged to provide some reports, which we found that not all even does that. So, and the last thing, so at the national policy level, these efforts are being made sometimes to incorporate environmental and biodiversity provisions as part of environmental policy, but not agro policy. So this is also something we're going to look a bit deeper and provide some recommendations for regulators.
Right. And then I want to close the presentation with some preliminary results. So our study is very much ongoing and there's nothing solid yet, so please take this with a big grain of salt. But this is some analysis that my master student Lisa Kallström did for her master thesis. And I won't go into details on how she exactly calculated this, but she actually found a somewhat surprising result that plowed fields had a higher climate resilience of pest control communities compared to conservation tillage fields. And one hypothesis that she has is because the body size of the animals that occur in these fields is quite a lot bigger, and she assumes that they consume more pests that smaller predators do. And one hypothesis she had linking to this is that communities that occur in heavily managed fields are themselves already quite resilient to disturbance, otherwise they would not occur there. But yeah, stay tuned for more results from our study. We have some next steps, and that is maybe to analyze the data collected last year and from the field seen this year. Next year, we want to expand on the microcosm experiments to test the hypotheses on community level. So we want to look at the evenness and community composition of the predator communities, how this affects predation rates. And finally, we need to synthesize the data and for recommendations for the future, where we hopefully do not need to rely on synthetic pesticides as much, and rather we want to capitalize on the assets that nature provides for us. And finally, I want to thank you for listening and our consortium members that contributed to this project, and yeah, the beetles for providing our data. Thanks.
Thank you. Are there any questions? Ladies first. Sorry.
So are you characterizing the landscapes? Because I can imagine, for instance, the size of the fields, if they have or not hedgerows, lines of trees that include the arthropods or not.
Yeah.
This is having also a very big impact in the...
Yes, this is an ongoing discussion in our project, how exactly we want to characterize the landscape, which we want to consider semi-natural habitat, so to say, in the surrounding landscape, but that looks very different in Italy than it does in Sweden. So yeah, we will characterize the landscapes. How exactly we will do this is still a topic of discussion.
Because it's a big covariance of
Yes.
the impacts.
Yes, we totally agree. And from the existing literature, we know that this has a really strong effect on which biodiversity even occurs in the fruits. But it's happening, but I have nothing to tell you about it, sorry.
We will follow you.
Right. Next.
I'm just really curious how you induce puking.
Yeah. So we select the beetles alive in traps that contain wood chips, so that we hope that they don't eat each other too much, and we put them in individual Eppendorf tubes. And these carabid beetles, they puke when they are stressed, because it is it has a very strong smell. So if it's caught by a bird, for example, it will often throw up to try and hope that the bird doesn't eat it. And so basically, in order to mimic this, we dip this Eppendorf tube in hot water, and the beetle will throw up in the Eppendorf tube. Then you can take the beetle out and release it, and then extract DNA from the
So using fecal samples is not an alternative for that?
Well, I'm not sure. I think the benefit of using gut content from the stomach is that it's a more recent consumption of like what it ate most recently, and that you don't have to wait for it to, otherwise we would have to keep it until it poops, I guess.
Yeah.
I don't know. This is the practice in this field.
I know it from bats where you take
Yeah.
the bat shit literally.
No, I think this is just a very quick way to make this happen. I don't know if there are more humane methods to do this, but at least we don't have to kill them. So then, I don't trade off for the beetle, I guess. Yeah. Are there any more questions?
Good.
All right.
If not, thank you very much. Can we make a little change? Sonoko, can I ask you to maybe do the presentation before, and then because I have the other one on the stick. So we continue with the project. Leah. Presented by Sonoko Bellingham.
Thank you very much. So I'm Sonoko Bellingham, and abstract, and there are two persons. I'm representing both Sonoko Dorothea and Bellingham in a single person, and now showing you the result of our project CLEAR. We have 6 partners from 4 countries. I go to them later on again. And as one talk already on the landscape and the following another with the landscape, I hope that you take home that landscape is quite important scale. It's quite difficult to define what's landscape, but it depends. That is our conclusion, that it is not definable. Can be few hundred meters till a few kilometers. That is the difficulties of a landscape. Another difficulty is there are many stakeholders and varying. So we heard about the hedges, landscapes, field shapes, and this depends also with the scales. So we are tackling this scale because of the importance. You know, everybody that the influence of agriculture on the biodiversity that's mainly happening at the landscape scale, based on the simplified land use, as well as the habitat, the decrease of the habitat richness, which means diversification of the landscape, again in the landscape scale. And of course, it is not biodiversity that the species are getting less, but also the function, ecosystem services in various ways, and also the resilience. We already heard of the apples, that there will be also some aspect of the biodiversity concerning also the productivity. But it's not only the negative side, agriculture can also turn it around. If it is all forest, it will be also a different area with also, again, less biodiversity compared to this mixture of the open land use. So we need the agricultural land use and turn this to a more sustainable use, the ecosystem services in agriculture ecosystem with increasing biodiversity as well as this multifunctional functionality. It's an old term. I think it has been researched for decades, but still, again, it's important. And we are still not yet ready to address all the points. So in our project, we assess the current status of the landscape, and then again, further assess the impact of ecosystem ecological potential of existing practices on biodiversity and ecosystem services, and assess again the policy and the landscape scale, and also point out the future action. And then we come to the action points that we elaborate future land use possibilities through, this is an important term, collaborative, because in the landscape it's not only one user, and you cannot say, now you have to do this and everybody does the same, then it's again a uniform land use. It has to be diversified, so it has to be discussed. Collaboration is necessary, that it is still diverse and in a harmonized way. So that is the difficulties, and you can imagine with many stakeholders and also with the angry farmers now. We all know for the beginning of the year, it is very difficult, challenging, but it's very important. So our goal is to support biodiversity, also increase the resilience, so productivity is also very important for us, and have this multifunctionality in agriculture landscape to be realized. So we identified here 3 different stakeholder groups. We have it somehow differently shaped. Later on, we come to a more specified one, that we have these 3 areas. So one is this again, sustainable land use. So what kind of practices is necessary, that is in the practical way, but also scientifically varied, that we have defined what practice should be done. Then what incentives should be created by the policy, also by the scientific analysis, to identify which kind of measure enhance it and doesn't go to the opposite side. So some policy measure goes to in the wrong way, even though it is has to be enhanced. But like the German Heizungsgesetz, it goes the wrong direction, even though you want to increase sustainability and it buys gas. So the policy has to be incentive that it goes to the right way. So it is not only policy, but also has to be analyzed. And then indicators, again, the practice, the stakeholders, the user is necessary, that what policies, what measures, indicator is necessary to be used, to be accepted, and also monitor the outcome of those results. So this is a very complex approach, you already may have noticed, and our project is also very complex. We have only 4 work packages, 3 is the active one is the coordinating one, and there are many arrows going back and forth, and many members are joining in double affiliation in different work packages, but basically we analyze the indicators, like the more natural science part. In the work package 1, we select and validate the indicators for the agroecological measures. Then the participatory analysis of the stakeholder, how those measures are accepted or can be utilized or as a preference in various ways. So in various stages are analyzed here, rather the social science aspect, but you see here the quite interlinkage. And then it comes to the landscape impact, where it's an upscaling process. We are only small countries throughout all the Europe, but we want to have this case study, and also that's this way of analysis, and how we analyze the collaboration of those different parties can be also used for the other countries and other conditions. For the generalization, it's also important. So as I said, we are 4 countries. And so if you see the whole Europe, it's not covering all, but I said if you want to have this process clarified, that it is a use case and can be applied to any other conditions. So there are findings, it's not the key findings, we are just also in the middle of the process, and if you imagine that we are highly interlinked, we are not quite, it's moving slow, and later on all the milestones is upcoming, so now we have to get to the right end. And here's how we are proceeding. That is a catalog of the agricultural practices and ecosystem services related to agrobiodiversity. And here we have for each countries, we created this kind of catalogs, what kind of indicators, monitoring practices, units, level of measurement, source of indicators, and so on. And these are somehow also directly related to our use cases. So it is not a general literature overview, but studies and researches and directly developed for the specific area, and that already shows the challenge of our project, because there are even only the 4 areas. We have these different levels, different time of measurement, different method, different scales, and that if we are now struggling with this, but if we can conquer this point, it may also this upscaling process that it can be also applied to any other area, how to proceed. So one of this outcome of this part must be the user-friendly communication. So now it's totally not user-friendly, so I think you cannot read it. I also doesn't mean that you can read it, but it's just I want to show the approach, and it's even much more longer. Then another one is our maps for the use cases. We call it landscape window. So as I said, the landscape is such a diverse scale, and it depends on how the aggregation, resolution, and the scale. So we said it's a window. You can, if you see out of the window, if you move, the scene also changes. But you can set as a window and analyze it within the window, this aspect, and you can imagine that if you move it, it slightly changes, but you can use this method for all other landscape scales. That's why we can take the landscape windows. It depends also on the site, that it here is the same scale of 5 by 5 kilometers, but according to the study site, we have to include the river and the other part, so it also can change, the window size can be also changed. This is just the first result of the land cover maps. And again, here there are differences about this resolution and scales. And now we are thinking rather to include another way to analyze this one, to have different resolution, like EU level resolution and local resolution, expert judgment based. Because if here is just a wheat field, they can include a flower stripe, but it is still a wheat field. It doesn't appear. And so, but the expert knows that there are somehow how many percentage of the area will be also implementing flower stripes. So we have to do it with a final resolution, but also scale up at the EU level, and that will be our challenge now here in this area. And not only the land use, that it will be the base for analyzing the ecosystem services. We used pollination as the indicator of biodiversity, carbon stock, and soil erosion are one other ecosystem service we include, and habitat diversity again as an indicator of biodiversity, and crop diversity to have indicators of the biodiversity. And this will be again visualized to show the hotspot and cold spot for those ecosystem services. Carla, who did the pitch in the plenary, is now analyzing the stakeholder 42. We got the interviews face-to-face, direct interviews with those different stakeholders. And so we have to can reflect the acceptance and the preference of those different measures and the importance of some measures for the ecosystem service based on those interviews. So reflect their need and preference on the measure, work on collaborative incentives to enhance biodiversity, because this collaborative is part of the most challenging one to. And surely, as I said, these are the time what we have to deliver to PTO and all the other holders. It's almost all upcoming now. So it is now, we have to know, it's now the time where the fruit is ripening, and now we are hard working to go to this report. So the next step toward impact is after we have those maps and measures, this get a broader, higher societal attention would be necessary, and inform policy makers of the result to move to the next agroenvironmental policies, and dialogue of the co-design and opportunities between scientists, landowner, and policymaker would be necessary. And for feedback for us for the next in the evening, maybe is as one of the stakeholders, a scientific stakeholder, if you have some advice to us how this format of this information, how to get the impact, will be web map, catalogs, scenarios would be quite, if you can share your experience for having attractive result offering for the community would be quite appreciated. Thanks a lot.
Thank you very much. Is there a question? Yes.
Two short questions. Could you name one or two agricultural practices that you were talking about?
One of the can be hedges or flower stripes, also reduced tillage can be also one measures. In areas with in the Scotland, we have partners where they have animal sheeping grazing areas, so the free sheeping frequency.
How many of them did you list already? Do you know?
So the listing was based on what is being done in the region. So as we said, it's not a literature review, it's what we've done. And then we reduced to see what are the common ecosystem services on them, so that we can focus on that are most common for all across all the regions, not the practices themselves, but the ecosystem services they're benefiting or they are generating.
I don't remember 30 to 40.
In total.
And then we use that.
And these land cover maps, you developed them based on expert knowledge, you said?
We have different maps, and this is also depending on the area differently. In Poland, we made the drawing by ourselves, and in Scotland, they have their maps, and so the resolution are different. But we want to compare the size, so we have now thinking that a map and resolution map, land use map available for the EU, total EU, Europe, using this resolution, and this local maps, also some kind of satellite maps, and also drone maps, and so the project maps also, and then there some expert judgment, and then compare this low resolution EU-wide maps with the expert judgment maps, and analyze what is the discrepancy, how can be, because you cannot have this expert judgment from all the area, so you have to somehow, bridge this gap.
No, I believe sometimes you cannot notice from the remote sensing the practice in the field. So
This is our challenging part, and now we identify this challenge, and we want to do both because we chose this use area because they are expert knowledge, they are research already done, and then people know the area. And on the other hand, we have only the map definition, etc. Thank you.
I am wondering whether you connect your landscape analysis with productivity typically on disease epidemiology. Some colleagues of me work on impact of landscape on disease in field, and I think it will be very interesting to connect your analysis because I think a good way to convince farmers is to show that landscape have positive impact on yield, on disease, on chemical consumption of.
Yeah, that's a good point.
You integrate this kind of things.
Yes, those areas are somehow unfortunately not in this window, so they are more in another area. But we want to try also to include this part and also part of the integrated pest management assessment. So that's why it will be also interesting and to go to the farmers. Of course, pest is one of the most important parts to, yeah.
I know for wheat, the pest management on the pest disease, there is project on which they use European database on landscape. You know what is the impact of on some disease, I think. It's available to connect.
Yeah, we will do your thanks, yeah.
Good, okay. Thank you very much. Okay, that's all.
Good evening to everyone. On behalf of our project manager, Professor Eligio Maluza, who couldn't attend today's meeting, I would like to give a short presentation on our Agrobioconnect project, Connections in the Landscape, Role of Landscape Complexity in Agro-Ecosystem Sustainability. Several European countries are involved, apart from Poland, which is a leader. Also Finland, Sweden, Denmark, United Kingdom, and Italy participate, performing quite diverse research tasks. For example, the English team focuses mostly on crops under cover, for example, in greenhouses or polytunnels. Italians work on spider mites in grape cultivation. The Swedish team tried to prepare a special tool dedicated to farmers to spread the results that we have been obtained within our project. And in Poland, we do a bit of everything. So, after previous presentations, it should be obvious that a greater landscape heterogeneity translates into more biodiverse and resilient agroecosystems. For example, semi-natural habitats in surrounding area of crops providing potential insects such as natural enemies of pests with shelter and with other resources, making their populations stronger, more resilient. However, many mechanisms underlying are still unclear, and we believe that our project, as performed under different European landscape conditions, will shed more light in this issue. So, the most important thing for a farmer is how to put knowledge into practice. And that's why we would like to take a look at the correlations, relationships observed so far between landscape and ecosystems from the practical side. We want our work to help fruit and vegetable growers to select, to choose the best and most sustainable agricultural practice in particular landscape conditions. And our objective is to link the landscape complexity over time and land cover dynamics with the above and below ground ecosystem services. Because, as you know, I'm into research on evaluating some biodiversity enhancing practices. And what I have learned so far is that obtaining very good results in one location, I mean lower pest pressure and higher beneficial insect activity, means really nothing when you omit the landscape impact. We need to consider landscape conditions in choosing the best agricultural practices. And how we approach our objective. So, we use the satellite remote sensing, which in brief, we take satellite images, for example, from Google Maps. We obtain them also by means of drones, which are later pixels from these pictures are divided based on their NDVI profile into different clusters, and the proportion of these different clusters helps us to describe local landscape conditions. And this satellite analysis are the background for our field work, within which we assess the below-ground biodiversity by taking soil samples and analyzing microorganisms, bacteria and fungi present, and assessing the proportion between different groups of microorganisms. We also assess system services and the arthropod diversity, applying both traditional methods such as pan traps in different colors, observations in transects, walks, identification of insect trap based on the morphological traits. For example, I focus on the parasitoid wasps. We assess the main pest pressure in our locations, for example, aphid pressure, codling moth, But we also apply one modern, very interesting method of assessing general entomofauna biodiversity. Using this tool, you can see this sensor, you can see in the second picture, which is the fauna photonic sensor describing insect populations based on their wing beat frequency. And our work is still in progress, so you need to wait for our results. But I encourage you to visit our website, where you will be able to find more information. Thank you very much for your
Thank you very much. So, any questions?
Yes. How accurate is this wing frequency analysis? Do you have any indication of the level
Not
of
yet,
accuracy?
because we started this year. We finished our work this season last month, so I still need to compare results obtained from these sensors with results obtained by me in the field using these traditional methods. But so far it looks quite reliable. This is some novel approach developed by a company from Denmark, I believe. Agrobiodiv.
Well, I don't think this is really a question. It's more a comment, maybe, and not it's only for you. Which, by the way, I think is very important that we have a more landscape view of our farming systems and try to, and what do I say? I'm going into the biodiversity things, into the agriculture system, and try to think about the ecosystem services and this approach. But still, I feel that maybe this is something for tomorrow, actually. I have the feeling sometimes we take for granted that having a more biodiverse system, it will, the ecosystem will perform better, or the agriculture system will produce more. And I'm not sure if all the scientific data are actually supporting all of this. I mean, biodiversity is important, but what about functional biodiversity? I mean, biodiversity per se might not just lead to a more productive ecosystem or agroecosystem in some cases. And I don't know if we are really looking at that, you know? I mean, it's not really a question for you. So it's just a comment from myself, I'm saying. And growers are actually asking themselves, because we here, sitting here, we like a field with flowers, with hedges, with trees, but then the growers are there and they need to produce. And they know that sometimes if you have catch crops, cover crops, they might compete for water, for nutrient. And at the end, those practices are actually going to be implemented by growers, and growers need to see if really their production is higher or not, or to cover crops, for instance, or flowers are reservoir for some predators, but are also reservoir for diseases sometimes, or viruses, for instance. And so I don't know if we have, and growers do know this sometimes, and that's why they don't often apply the things here we are researching. So you that you work at the land, now I'm going to a question, sorry. That you work in a landscape level and you are in connection, do you think this is just myself, I'm kind of little crazy, or you think that?
Yeah, I want to comment. Yeah. I think you're totally right. This is a diversification not per se, because the intensive system was developed to be productive. But the problem is that the focus is only on the production, so it's only producing wheat ton per hectare, and forget all the other production of the agriculture. So the underground water quality, carbon storage, microclimate, these are not valued. And only the wheat and the decitons are on monetary value, and so that the producer are looking at those values. And if this one is only valued, I think this system doesn't change. It will be the same simple big intensive system produce the most in this year, but not in the future. That's a problem. The resilience in the future, climate change may get much harder impact on it. Then this resilience that it's balanced, not the highest value always. That is, I think, the current situation, that you have all the measures inputs to get the highest value because it's economically valuable and the farmer has to survive. This is not a problem, it's a system. And if it is more resilient and then it pays off for long term, and also the other services will be also paid, then it's changed. And then this smaller system, diverse system has the advantage, but its economy is not yet there. Would be my, but we can discuss also later.
I think very inspirational comments. Thank you very much. I think leading maybe for the dinner more discussions or general discussions. Thank you very much. Thank you to all the speakers. Thank you for your participation. Thank you for the people joining us online. If there are no questions, we can close now. I can tell you from the meeting hall at the front of the I think they also want to take a picture. I think we have to be